Let's get into the pathophysiology of Huntington's disease. It is caused by an abnormal protein called Huntington, which is also known as HTT. So most Huntington proteins, they have a CAG repeat of 10 to 35. And for those of you who don't know what CAG stands for, it's cytosine, adenine, and guanine, which are nitrogenous bases found in your DNA. The three of these nitrogenous bases together are called nucleotide triplets, and they're used to encode the amino acid glutamine, which is used in the biosynthesis of many proteins. People with Huntington's disease have a CAG repeat of 36 or higher in their Huntington proteins. So this causes the structures of the polyglutamines formed to be abnormal and much longer than the average person's. And this alters the Huntington protein as the abnormal polytriglutamines cause fragmentation and aggregation. So for those of you who don't know what fragmentation aggregation of DNA is, fragmentation is a separation or breaking of DNA sequences from their strands into small pieces. And aggregation is where multiple chains attract each other and they condense to cause mesophases or other complex structures. This causes the HTT protein to become clumped and to misfold, and that causes them to be toxic and deadly to neurons. And this goes for all around the entirety of your brain, but as you can see here, it mostly affects your basal ganglia, seeing that they're much smaller now. And even more specifically in the basal ganglia, it affects the striatum. So the basal ganglia is a group of subcortical nuclei responsible primarily for motor control, as well as other roles such as motor learning, executive functions, behaviors, and emotions. As noted in the symptoms, Huntington's disease causes damage to the basal ganglia, which may cause problems controlling speech, movement, and posture. This combination of symptoms is called Parkinsonism, but for people living with Huntington's disease, these specific uncontrolled movements in those patients are called Huntington's chorea. The striatum is the main input unit of the basal ganglia, and it receives excitatory glutamatergic inputs from the cerebral cortex for synapsing pattern reflex to the topography of the cortex. And it's also really noteworthy to mention that dopamine is released in the striatum of the basal ganglia during human emotion processing. So this will often cause dopamine levels to lower in people that live with Huntington's disease, and can be a good reason as to why Huntington's disease has a comorbidity with depression, anxiety, and other depressive symptoms. And let's get into the onset and progression of Huntington's disease. It is caused by how many CAG repeats one may have. The greater number of the repeats in the Huntington protein, the earlier the onset and the faster the progression of it. The high amount of CAG repeats will also increase the likelihood of the inaccurate DNA replication. The repeating sequences can actually form loops, which causes the DNA polymers to add more repeats as it replicates the DNA. So what this means is that if somebody has Huntington's disease and they pass it down to their children, the CAG nucleotide triplets will be passed down at a higher repetition rate than their own. And then children who receive this disease will experience symptoms sooner than their parents will have, and they'll have more severe symptoms. This is known as genetic anticipation. People who have 40 to 50 repeats are usually experiencing symptoms in their 40s, but can experience them anywhere from their 30s to 50s. And people with 50 to 60 repeats will have what is known as juvenile onset of the Huntington's disease. And as you can see here in this next picture, I really like this because I think it describes the CAG sequence being repeated very well and just shows how the Huntington protein will become clumped and it will eventually be toxic to a neuron and it will die. And I also think that this picture is really good for describing the pathophysiology of Huntington's disease. As we can see, each stage in this, the transcriptional dysregulation of the CAG, how there's impaired protein degradation, There's altered protein folding of the Huntington's protein. You can see the disrupted neuronal circuitry, and you can see the mitochondrial dysfunction, which is where the neuron dies.